All right, what's up, everyone? Welcome back to another Ekkolápto Polymath Salon. Today, I'm joined by the Polymath Medical Salon special guest, Willem Nielsen from the Wolfram Institute, as he will talk about the computational foundations for medicine, which him and Stephen Wolfram, worked on a little while back. They also have a new article about applying cellular automata to innovation, which is very fascinating. I'm actually going to link that in the chat for everyone here. But without further ado, let's have everyone give a quick introduction about themselves. So, Willem, you want to go first real quick?
Yeah, sure. My name is Willem. I work at the Wolfram Institute as a research fellow. My background is in computer science. I did my undergrad at Vassar College. And yeah, I'm just really excited about computational biology and just really happy to be here. So thanks for having me.
Great. Misha.
You guys probably all know me. I'm a member of the MPCR lab, and my research is primarily on the AI side.
Oh, you guys probably know me, so I'll try to keep it brief. I'm also a member of the MPCR lab, Ekkolápto, and a affiliated researcher with Wolfram Institute. My main area of research is general relativity. I also research complex systems in general, and I'm a PhD physics student.
Hi, my name is Adar. I work at FAU, but also in England at University of Cambridge, University of York. I develop medical technologies to treat different mainly neurological and movement-related conditions, and also attempting to understand how the brain works in a very general sense.
DVZ?
Howdy. I am also in the MPCR lab with Misha. I am a PhD student, soon to be candidate in a couple of weeks. And I have a number of research projects I'm working on, but the wider theme is making AI smarter to make humans smarter.
Wonderful. We also have a new guest, Sergey, if I'm saying your name correctly. You're very welcome to give a introduction real quick.
Hi everyone. Yeah, I'm excited to learn about this. Was intrigued by the name. Also, I'm interested in computational medicine. I'm working I'm a founder of a startup for health coaches, and I'm like a biohacker since maybe it was last 2012. And last year I was reading the Active Inference Institute Discord and following this topic. So that's where I found this and learning what you do here.
Wonderful. All right, we'll be getting started right about now with Willem's presentation. All right, take it away. Go for it.
Yeah, thanks, Addy. So Stephen Wolfram and I worked on a blog post a couple months ago now about trying to build computational foundations for medicine. So the idea is, as it's practiced today, medicine is almost always about particulars. This has gone wrong, this is how to fix it. But might it be also possible to talk about medicine in a more general, more abstract way, and perhaps to create a framework in which one can study its essential features without engaging with all of its details? So again, we want to emphasize that our goal is not to create specific actionable conclusions from the model, but instead it's to try to take these general phenomena that practitioners observe and talk about and try to make them more precise to help build people's intuition in the field. So our model is based on, this is the model organism, and it's a cellular automaton, which K equals 4, R equals 1, which just means that there's 4 colors, and then there's 3 boxes that affect the rule below it. So the way this works, if you're not familiar with the cellular automaton, is basically just you have, for example, if you have 3 yellow cells on top, then the one below it, the one in the center below it will become yellow. And you just use all these rules and update the system, and this is the resulting pattern that you get for this particular rule. One important thing about this is that we kind of use the length of this, or how long it quote unquote lives. So until it gets to all white, the number of steps until it gets to all white, as the kind of the goal of the organism, or what it's trying to do, the high level goal. And then the pattern of how it gets there is kind of like the mechanism for how it achieves its goal. So that's the idea. And so the whole point of this post is basically to do perturbations to the cellular automaton. So when we draw an arrow, it just means we're changing a cell at that location. And then we observe what happens to the pattern as a model for disease. So it's like it's like you poke like a tadpole when it's growing or something, and you see what happens to how it like ends up looking. So the important thing here is there's different, you can already see there's different kind of types of disease in our model. So first of all, in this first panel right here, you have a perturbation that didn't affect the outcome of any of the details really of the organism. It kind of like changed a little unimportant thing, and then everything went back to normal. In the second one, you can see something where the goal of the organism is still achieved, but the way it does it is changed based on the perturbation. And then the other ones are examples where the perturbation is kind of malignant, or it prevents the automaton from living 101 steps and achieving its goal. And in particular, this last one is interesting because it's like basically what's happening here is there's some change that causes some like subroutine or sub-pattern within the organism or within the automaton to grow too much, and it's uncontrollable, and so it's kind of like we call it like meta cancer, or it's a meta model for cancer. Okay, so that's the disease, but then the question is, can we once we've made a disease perturbation, can we put it back? Can we get the organism back to its original state? And that's our model for treatment or medicine. So these are just different examples of single cell perturbations trying to get the organism back to achieve its lifetime goal. And you can basically just see that some of them works. So like this middle one right here, you were able to perturb a cell and put all the details of the organism back together. And then in other cases, you change the cell and it doesn't work, or you are able to help the organism achieve its goal again, but the way in which it does it is changed. Okay, so the first kind of thing we looked at was the classification of disease. So in Western medicine, it's kind of a tradition to put diseases into buckets. So like, this is heart disease, this is lung disease, et cetera, et cetera. And the idea is, what we wanted to see is, can we do that with our model? Are there such a thing as these discrete categories of disease? So on the left here is just a bunch of random perturbations, just to give you a sense of what happens when you perturb the organism. And you can see a lot of times it doesn't change much, but then we have cases where it stunts the growth of the organism, so it shortens the length of the automaton, and then cases where it makes it grow too fast. And over here is kind of a mapping of a sample of all the possible diseases in feature space. So we're basically just giving a machine learning algorithm the image, and it's trying to categorize based on that. And so like you can see down here in this like tail, you have the shorter automaton, and then over here you have like longer ones. But the point is that they don't fit into nice like clusters, so they're like one disease kind of like melds into the next disease. So the idea is that it's suggesting that this nice discrete categorization of disease that we kind of assume is possible in medicine may not be possible, and it's a function of the fact that of computational irreducibility, where you make some change and then there's like so many different ways that things can go wrong. Yeah. Okay, so after we looked at classification, the next thing is diagnosis and prognosis. So kind of our model of a doctor is someone who is using high-level metrics of an organism, or in our case, an automaton, and trying to classify what's happening to it, and also make predictions about what's going to happen to the automaton. So basically what we do here is we're looking at a high-level metric, which is the width of the organism or the automaton. So basically, if you look at this picture to the right, the blue line there is like a growth curve of the organism. So you can see it's increasing, its width increasing, and then as it's about to die, it decreases the width, and then it goes to 0. So, and what all the orange ones are all the possible diseases and all the possible growth curves. And so basically we're saying, like based on just a high level metric, can a a computational physician be able to predict what's going to happen to the organism when it's going to die in this case? And so what you can see already is that it's just not clear when, like there's just a lot of noise in which patterns make the organism die when. So already you're seeing it's kind of confusing. And so what we did, the question is, well, one interesting question is, can AI help with this problem? So basically what we did is we took a width history. So we took like a segment of that, of these growth curves, and we gave it to a machine learning algorithm. And then we asked it to predict based on that width history, the lifetime. So it's kind of like a patient giving the doctor their history, and then and then the doctor then has to predict, whether, that patient is going to, what's going to happen to the patient. So, and the question is, can AI help? What you can see from, so what this is basically just like a comparison of the predicted versus the actual. And if it was really accurate, then the whole thing would look kind of like a diagonal line, which is what you want. But what you can see is once you, like from 40 to 60, the predicting those lifetimes, it can kind of do it, and it's a little bit noisy, but it can kind of do it. But once you get past that, then it becomes really noisy, and it's really not able to it's not able to tell what's going to happen to the organism. So it's kind of showing a case where the AI model doesn't have enough information or to make a prediction. So it's not that it's not that doctors aren't smart enough in this case. It's just that the data is not there. And I think this is really, like, I don't know, it seems to ring true. Like my dad happens to be a doctor, and I was asking him about this yesterday, and he was saying like the patient, a lot of your information comes from the patient, what they tell you, and they're like really unreliable because so it's just you're standing on unsolid ground as a physician. Okay, so we talked a lot about classifying disease, diagnosis. What about finding treatments? So basically the above row is just showing random perturbations to the automaton. And so basically the point is that most of them don't actually heal the disease, but you can see in the bottom row is all the ones that bring the automaton back to its original length. One question is, so this is kind of all single cell treatments. And importantly, the bottom panel, a lot of them don't, all the details of the organism don't go back to the original. One question is, what does the space of all treatments look like? So basically in this picture, we're looking at a single disease. So this is the original organism, and then to the right of it is a disease. And then we're looking at what happens if at each cell, let's see what happens if we change that cell as a potential treatment, and how effective is it? So it's kind of like a therapy sensitivity plot. And so if it's dark red, that means that the therapy made the organism live too long. And if it's light orange, then it means it made it live too short. And if it's orange, that means it was an effective therapy. And the point here is that the mapping of where you should give a therapy is not at all, there's not very much meaning to it, or it's very messy. So it's hard to tell. The point is, it's hard to tell where to give the automaton the treatment. And then ultimately it's a function of the irreducibility of the organism, because when you make a change to the organism, you don't really know, nobody knows what's going to happen, and you just kind of have to run the rules and see. So that's the idea. So we've been looking at like single cell treatment, but can we do better with like different types of treatment? So in this case, we did an adaptive treatment. And so what this is showing is basically in the first panel, we have a disease, okay? And now we're going to try successive treatments and keep the ones that get us closer back to the original lifetime. So like these first two panels, they gave a lifetime of 61 and 65, which wasn't closer, so we rejected them. And then this third panel, or the fourth really, is it did a treatment that made the automaton live longer and closer to the original lifetime, so we kept it. And then we just keep going down the organism like that. And so with this approach, it's much easier to get it back to the exact lifetime. It's much more effective. And so you can kind of imagine like a case where, you know, you're able to do multiple treatments of a patient, or like try different drugs kind of thing, and just keep what works. And is, you know, our model suggests that that is an effective approach. Okay, so now we can even do even better by being like doing a more aggressive treatment and finding a perturbation at every single cell. And it's kind of like slowing time down. And it's kind of like an analogy to the fact that, you know, you kind of have to catch a disease early, or else, you know, irreducibility takes over, and then it kind of gets out of hand. Okay, so the last thing we looked at is genetic variation. So basically our model, it's nice because this particular automaton has some quote unquote nascent genes or rules that it doesn't actually use in the original pattern. So those are highlighted in yellow here. And the point is that when we give the organism a disease perturbation, then those nascent rules can become active and affect what happens with that disease. So it's kind of like looking at what happens to a population if they all get the same disease, but they have, you know, they have the same phenotype, they all have the same pattern, but they have different genomes. So is it true that they're all gonna, you know, what's gonna happen if they all have the same disease? And so this is basically what that's showing, is a population response to all the same disease. So we do some perturbation, and then we look at all the patterns we get for this, like all the different possible gene combinations with the same phenotype. And so basically the point here is that you can just see there's a lot of variation. And that just because, you know, some disease affects one automaton one way doesn't mean it's going to affect a phenotypically equivalent but genotypically different automaton. And so then it's cool because then you can ask a question, you can kind of do like a computational clinical trial. So basically we give all these automaton the same perturbation, the same disease perturbation, and then we give them all the same treatment, and then we ask what happens to different gene variants. And so what you can see here is there's again just a lot of variation. So it's kind of like if you know, so in this fourth panel from the left, it treats the organism pretty well, and it brings the automaton back to, you know, its original lifetime. But in the other case, but just because, so basically it's like just because a treatment works for one, you know, set of the population doesn't mean that it's going to work for all of them. And it's something that, you know, you see in medicine. To try to kind of give a conclusion and takeaway here, basically the fundamental idea is that, you know, when you make some change to an organism, because of computational irreducibility, it's not clear what's going to happen to the organism, and it's not clear what to put back or what pieces to put where. It's kind of like trying to unscramble an egg. And so it gives an intuition for why medicine is really hard. And it also kind of gives you a framing of what it means to, what medicine, like what the goal is and how to improve it, which is basically to find, you know, pieces of reducibility where you know what's going to happen to the organism, and you're able to use some model, whether it's an AI model or human intuition, to figure out where to treat the organism. And also, you know, the other side of that is, you know, you can kind of recognize that you don't know, and just like in the adaptive treatment thing, where we just try a bunch of different drugs and then you know, see what works basically. And so it's like finding those situations is kind of the goal of medicine. I mean, Stephen Wolfram kind of like had this idea, and I just like helped him with it and helped him develop it, and it was just like an amazing experience and really fun. And so thank you, and thank you to you guys for listening.
Awesome. Thank you, Willem. That was very fun. Awesome. This is actually a great time to maybe bring up, there's a question that I wanted to bring up. But before I do, I wanted to know if anybody else had any questions for Willem before I go down a rabbit hole.
I do.
Go for it.
This is maybe a general question, but so a lot of times when you do some very theoretical work, you put a section at the end of the paper that says future directions on where you could see this going in the future.
Mhm.
Let's say somebody thinks this computational approach to medicine is a really good idea, and they give you $5 million and a research team of 20 people and whatever to explore it further.
Mhm.
What directions would you be interested in taking this research to get it further down the road?
Yeah, so one thing I was thinking was, I mean, I really want to see how much reducibility is in our model. And because we the machine learning thing that we tried was basic, and it was a small model, and we only gave it the width history, but we knew that there's a bunch of other things to try, but because like you just kind of we're just trying to finish the post, we didn't really get the chance to. But yeah, that's what I would do, is try to figure out, is it really true that the AI model can't make accurate predictions? And I would suspect that basically you're just going to hit some limit where computational irreducibility kicks in, where it's like the only way to figure out what's going to happen is to run the program. But I also suspect that there's a lot more opportunity to make predictions with a bigger model, with better data, with a better interface.
If I could ask one quick follow-up question. So you told me about how you would advance the research along one direction, which is sort of the pure theory. What would you do to advance the research along like the more practical direction of taking this and figuring out a way to apply it? I know you said at the beginning that this isn't the goal of this research, but let's say you get $5 million and a bunch of people, and then NIH says, make this practical, Willem. We really like this.
Yeah, I would do, I mean, I think that there's a lot of opportunity where basically like doctors don't have the time to always look at scientific papers. And so there's opportunity where it's like you have some case, or a patient comes in and they have some set of symptoms, and basically you should have an AI model that's read all the papers that are relevant to that system and be able to offer a suggestion. And so, maybe there's some potential there with like basically having the AI be like a PhD student who's read all the read all the literature. And there's some people working on this. Like I remember Stephen was showing me something called open evidence, which is kind of, I think, similar to that idea. And then the other thing is, like I saw some case where someone had a rare disease that was lethal. I think it's called, Castleman syndrome. And basically what he did was he just used LLM, and he just looked up all the drugs that were used in diseases similar to his that were FDA approved, and he didn't even go to the doctor, and so he just kind of like started trying these different drugs, and eventually found one that worked, and so he was going to die otherwise. But I think like finding cases like that where you can actually like you have the leverage, or like to kind of take some more risk, even though you don't really know why something works, just be able to try different things, like something like that, if that makes sense.
Awesome. Thank you. This was a great presentation. I'm going to shut up, but I am also going to send a paper in the chat that I found while you were talking on someone who did similar work with attractors and perturbation, but with intracellular networks, that I thought was really interesting and relevant.
Yeah, by the way, I wanted to say I'm reading that book, Peak, because I feel like it's relevant to actually, like, we've been doing a lot of adaptive, like in evolution, but trying to figure out how that relates to like how humans learn things and how humans become experts. I think it's relevant. So thanks for the suggestion at the conference.
Yeah, absolutely. I'll cede the floor now because I see many other hands up.
All right, Emma, you're next.
Awesome job, Willem, by the way. Loved your presentation. I'm actually curious. So I know that you changed the automata in different ways to see how like different organisms would, I guess, be affected by different diseases or the same disease. But I'm actually curious, like, yes, there's so many things that we don't know about the disease and how it like progresses and all of that. But I feel like what's important in medicine are the unknowns within the actual organism. And I'm curious how we can go about figuring out what those differences between the organisms are in order to see how the disease might affect them, if that makes sense at all.
Okay. I mean, one thing I could say is if you were trying to, like, if you're really trying to dig into the details of this particular model, there are, like, you could think about it like there are certain rules that have different functions, and also there are certain rules that are used at different frequencies. So, like, to give an example, in this case, in this particular automaton that we're using, there's this there's this rule where one purple and two whites becomes a white, and two purples and one white become a white. And what that does, it has the effect of like it makes the organism stop growing. So if you look at this, I don't know, can you guys see my cursor?
Yep.
Yeah. So if the rule is kind of like it's kind of like apoptosis, or it's like it's making it so that the automaton stops growing. And so I've wondered if you can, like, basically, like, certainly if you change that rule, then it's going to be really bad. And then there's other rules that you can change that you probably can predict are a little more benign than that one. But I'm not sure if that answers your question.
Somewhat, but I think, sorry, and anyone can go if they really need to, or I'm actually,
You're fine. You're fine.
I'm more interested in, like, how do you know what rules? Everyone's going to have slightly different rules, right? Like, overall, we behave and we function as the same organism, but everyone has different genetics, everyone has different susceptibilities to different things, and I'm wondering how you find the rules in order to see computationally how we'd be impacted by the disease.
Yeah, so I mean, that's what we're kind of trying to do, is basically, like, I mean, the point is you kind of don't know. That's
Right.
the point, is because, so what the red arrow is showing is basically it's showing the first time that a inactive gene gets used in a disease.
Mm-hmm.
And I mean, you can just see from this picture that, like, for example, this last frame, it's like it's really hard to predict just because of that single change in the rule, that you get this whole new pattern. So it's like you really don't know how different different genes are going to respond in different ways.
All right. Thanks, Emma. Next, we have Misha.
So my question is about how this scales, actually. So I'm wondering, have you tested different sizes in terms of, like, state space of the genome, and what's the scaling law? How does that compare with human scale or other known organisms? And what can that possibly tell us about both the cross applicability of this work, but also about that reducibility question that you were asking earlier, posing?
Yeah, so in this particular automaton, there's only three rules that aren't active. And so because of that, it's basically there's 64 automaton with the same pattern, but have different quote unquote genomes. But we found cases where there's, like, there's way more inactive rules. And so there's, like, we found, I think it was like, there was like one with 64,000, a population of 64,000. But we just ended up using this one because it's nicer in other ways, like how it looks and stuff. But yeah, I think it's certainly possible to get something with a larger population, if that's what you're asking.
No, it's not that. It's how do the effects scale as proportional to the variation size, if you will, or the log of it. As in, you have a certain number of effects, right, that is as a result of variation between different genes, right?
Mhm.
And some of them are more significant than others. What is the law of scaling as far as the significance of certain effects and the effectiveness of some computationally discovered techniques as the number of possible variants changes?
I have a similar question about scale, just maybe not horizontal but vertical. So cells make up systems, right? So nervous system, like some other systems, and my intuition is that yes, we cannot kind of predict like the state of every cell or like reduce it to some formula, but we on getting higher and higher, there are like systems to self-balance, and even not knowing like what's going on the level of cells, we can predict what's going on with the nervous system. And getting higher and higher, we predict like that. Okay, this is if we drink coffee, then we stimulate the nervous system. So it's very similar. Like we can reduce all this complexity of the body to like stimulate in, result out, right? And so it's in the active inference, it's a concept of market blanket that we can consider the nervous system as a system with Markov blanket, where we don't need to know what's going on under it. It has an interface with other systems, and we just need to know the interface of the system.
Yeah.
Or like of the heart. We can know that there is the interface of the heart, and like and so my question is did you consider that the reducibility is on a high level?
Yeah. So I agree with what you're saying, which is like the idea that you know, and you can kind of see it a little bit here, which is that basically this organism is trying to achieve some goal of living a certain number of steps, and to do that, it's using different mechanisms, a lot of which are really complicated and hard to understand, but you can imagine that like because it has a goal, or it also has, like, you can interface with it at a higher level. And I don't know, how that fits into our model, I'm still thinking about, like how could we think about this organism as having is making decisions. I don't know if we could even have a model for that in this case, but I do think that what you're saying about, I think like it gives you a good intuition for the idea that there's parts of the organism that are simple. So like in this automaton, the end is kind of simple, where it's just like a big purple blob and it's just closing. But the beginning is like it's like really hard to understand. So yeah, I mean, I would just say that we're not saying that it's all impossible to understand. We're just saying we're just trying to give like a qualitative description of what that kind of looks like.
Well, thanks first of all for a really interesting, elegant presentation, and the model is very elegant, and it looks to me quite internally consistent, based on the rules that you've set up and so on. And I guess my question is, as someone who works with clinical data and with patients from time to time, how would you go about validating something like this? Would you take, for instance, a condition and decide on what the inputs and outputs of that condition would be? Like we, for example, working on long COVID. So could you take a single condition, say, model it longitudinally? And how would you go about that kind of mapping? I know this question sort of overlaps some questions that have been asked by others as to how would you bring it into the real world. But do you have any thoughts about how you would actually manifest this by having real clinical data and then testing predictions and so on?
Yeah, I mean, one thing I would do is, like I said before, which is we used this population because it had a nice, it had other nice features, but there was populations that we found of automaton that were much larger. And so like one thing you could do is just look at the histogram of, what is the lifetime distribution of, that particular population, and how does it compare to an actual clinical trial? You know what I mean? And another kind of dimension of relating it to the real world would be, so there's this thing called the ICD-10, and they actually have this kind of hierarchical classification of disease. And so what you could do is try to be more thorough with the model and try to literally make the exact same tree and kind of study the properties of the computational classification tree and then the properties of ICD-10 and see if, what the overlap is there.
Yeah, so maybe that'll help you characterize some things, and there could be predictions from that. But ultimately you want to reach outcomes, right? You want to have a certain clinical outcomes,
Mhm.
treating the disease effectively and so on. So how would you go into the nitty gritty of that? I know that you could use ICD-10 codes to
Mhm.
characterize the sets of symptoms or
Mhm.
descriptions of conditions, but it has to be relevant to a patient if it's going to be applied in that way.
Right.
So how would you go about that side of things?
Well, I think to answer those questions, we would need, the model is too simple to try to address specific, disease. Like this guy, he obviously doesn't have a heart, you know what I mean? But so I think we would just need to think about how to make the model more realistic, which would inevitably make it much more complicated. But yeah, I mean, we really haven't explored that, but it's interesting though.
I mean, maybe not. It might not necessarily make it more complicated if you got the right kind of mapping. If you found certain collections of rules, say, that would predict a particular condition. If you narrow it, say, to one condition, let's say, and just analyze, complex enough, but just analyze it sufficiently in detail to test drive it, as it were, using the
Yeah.
model. Just a thought.
Yeah. Yeah, I mean, we were thinking about like, what is the analogy in our model to a drug treatment? And we were thinking that it's more like when you change a rule, because it's kind of like changing some low-level computational process that happens a lot in the organism. So, and then like what's the analogy to something like amputation? It's basically like doing some treatment where you take some huge block of the automaton out. So, I mean, I guess there's something there where we could try to be really precise about that and say this particular type of therapy corresponds to this drug treatment, I guess.
Okay, interesting. Just one quick thought. Some conditions are sort of, in a sense, self-referential. So your knowledge as a patient of your condition can affect your condition. You have placebo effects. You have, the mapping of your body with amputations and phantom limbs and things like that. Can the model capture almost a recursive element in any sense like that might help the model situations like that? It's weird. I know, but
No, I don't think so.
okay. All right, thanks.
So throwing diseases into sort of a space like this to form a mapping is really interesting to me. I have an idea and I want to get your thoughts on it, Willem, and tell me how close this is to kind of something your model could do. So you have what's called embedding, which is very common in machine learning, where you throw things into hyperdimensional space. And one of the interesting things that your model does is, because it gets away from categories and gets towards hyperdimensional space, instead of saying this patient's illness fits in this category or this category, you could just say this patient fits in this point in hyperdimensional disease space and leave it at that. You don't have to categorize them. And then drugs also exist in a hyperdimensional space. So there's what's called off-label uses for drugs, where people get the FDA approval and everything for a drug for one use, but doctors use it for another thing all the time. There's some drugs where the larger percentage of their use is off-label. So what I'm thinking is, in embeddings, you sort of take multiple hyperdimensional spaces and try to match them to each other. Do you think that if your model was much more advanced, you could put a patient into hyperdimensional disease space, look at what drugs are close to that point in hyperdimensional disease space or other kinds of treatments, and sort of use that to decide the treatments for the patients, as opposed to more discrete category classifications? Does that make sense?
Yeah, that makes sense. I think that's exactly when you ask what I would do, that's kind of something, that's basically the idea, is to try to, I mean, when we only gave the machine learning algorithm, we only gave it, here, let me show you. We only gave it a width history. So we didn't even give it the colors of the cells or anything. And Stephen Wolfram has looked at similar things where you basically just give a machine learning algorithm a cellular automaton, and then you ask it to predict what's going to happen. And so you're kind of saying, we give the machine learning algorithm all the bits of the cellular automaton, and then we ask, where should you apply a treatment to this guy, and which color should you change? And we actually tried something similar. Let me show you really quick. Okay, so this is an attempt at that, where basically the blue arrow is a disease, and then the red arrow is where the machine learning algorithm that we trained thinks it should perturb to put the disease back. But basically, this particular model with this particular data wasn't able to do very well. So like this is kind of the lifetime after treatment. And so it should look like a peaked at 101 if the treatment is working, but it's all spread out. So it wasn't really able to find that particular treatment. But like I said, maybe it could do better.
So would it be accurate then to say, and correct me if I'm wrong, that a long-term goal of this way of modeling disease could be to get away from more discrete categories for disease in medicine and get to a more individualized place where a patient comes in with symptoms and we don't even worry about putting them into a box and then seeing what treatments fit in that box. We just look at sort of the symptoms and we choose the medicine based on that. Would that be kind of accurate to say that that could be a long-term consequence of this research? Yeah,
maybe.
Very long-term, 30 years. You're an old professor.
Yeah. I mean, I'm unsure about, how well that, it seems like a good idea, but I'm not sure how well it's gonna work. So I don't want to say, but I think it's a good, interesting idea.
This is fascinating work. Thank you.
All right, my question, and by the way, you are very welcome to speculate on this as well. But the thing that I question about a lot is like, what are these goals? What is a goal in a biological system, in a genital system? And how is that goal actually derived or generated or found, right? Because, people will say that, when an organism, say, loses its goal, that's sort of what we call the process of aging and disease. And there's sort of a dysfunction that's happening because it's lost its sort of goal or purpose of its cells and the organization of that. And so I get my question is what is a goal? Like, how would this article sort of identify what a goal actually is, and how is that goal found and generated by the organism?
Okay, so I think that's a reasonable question. And basically, we are defining the organism's goal to be. We're defining the organism's goal to be trying to live a certain number of steps. And when we evolve, so we found this automaton by an adaptive search. So actually, let me show you this. Basically, what we did to find this is we changed the rules one bit at a time, and we kept the rules that allow the organism to live longer, but not live longer or the same length. But the critical thing here also is that we also perturb the organism during its evolution in order to get it to be robust to perturbation. So this particular automaton is actually, out of all the possible automatons, it's incredibly robust to perturbation. Most of them aren't this good. So if you look at like the distribution of lifetimes, when you perturb this organism, it lives for 100. Like it's very much peaked at 101. Most automaton are not like that at all. They would be completely flat. If you change one cell, then the whole thing will blow up in most of them. But so what that means is, I mean, like you said, we're just defining the goal ourselves. And so maybe, you could come up with a model where you just set you build the situation where from the bottom up, the automaton or the model kind of automatically starts to try to achieve goals, if that makes sense. But we're just trying to keep everything as simple as possible. So in order to do that, we just kind of say, this is the goal and this is what you're trying to do, if that makes sense.
So Willem, would you say that, these things that we're calling perturbations are maybe a kind of stressor, right? Like potentially a good stress, a stress that kind of induces a positive, beneficial, adaptive, response from the organism. Would you say those are kind of the same terms?
Yeah.
All right, that's good, because then that leads me into my thing about hormesis that I love talking about, right? Because that's this big thing that people talk about it in the longevity field a lot, but I actually think hormesis has this very large application to just the way we look at information in general, and it's that I think that whatever things that the organism is interacting with is always a kind of slight stressor or signal for it. And there are thresholds that once you reach them, it actually starts to become, say, a negative thing,
Mhm.
right? So, you know, if you think about like a stressor or a perturbation or whatever, right? Like let's think about things that would normally kill an organism, right?
Mhm.
So we can think about starvation.
Mhm.
Starvation would kill an organism. Well,
Mhm.
how do you adapt to that? What you do is you break it up into chunks. We call that fasting or calorie restriction,
Mhm.
right? And then, you know, with exercise, if you over exercise or do something too strenuous, you can get hurt or kill yourself, right?
Mhm.
But if you chunk that up, that's, you know, what we call aerobic, anaerobic exercise, whatever.
Mhm.
And there's thresholds that we have for, say, undertraining and overtraining, reaching that,
Mhm.
you know, that sweet spot. And then same thing for even temperatures. Like, you know, you can freeze to death, or you can die of heat if you're exposed to too much heat for too long. And
Mhm.
how do you adapt to something like that that would normally kill or hurt you? Well, you break it up into, say, sauna sessions or cold plunges or whatever, right? And it's
Mhm.
interesting that whatever thing that the organism can't directly interact with, if you break it up into smaller chunks, it's able to handle it,
Mhm.
right? And now I also want to kind of tie this into like this bigger idea of immune responses and sensitization, because, you know, if you think about food allergies, now everyone here that knows me knows I love talking about this stuff, but food allergies, right? Somebody could be allergic to like a, you know, a certain nut or something like that, and
Mhm.
something that would normally make you go into shock and have trouble breathing and die, if you dose that up in very, very small amounts over a period of time, if you distribute that throughout time, all of a sudden you seem to not react to it in the same way anymore,
Mhm.
right? And that's just this big idea of desensitization and you know, adaptation. And so my kind of argument is that I think that what adaptive systems have are kind of built or evolved to do, through whatever way you want to say that happened,
Mhm.
is that they're trying to make the best out of a worse situation. Right.
Mhm.
So you could, I mean, dare I say, biology is inherently almost optimistic. It's going to try to make the best out of a worse situation. And that's just what we call adaptation. So I'm wondering, just kind of giving you these examples and exercise and all that type of stuff, what other applications you see for this model that you and Stephen have worked on?
Yeah, I'm happy you brought this up because I think our model actually has a really good like kind of conceptualization of hormesis. So basically, you can think about it like this, you know, this organism adapted with in an uncertain environment. So when we evolved it, we made many perturbations. Like, so these are all the green dots are the lifetimes after after giving it a perturbation during the evolution. And so, but the point is that, yeah, so like if you perturb this organism, a lot of times it actually is still, it's able to pretty much achieve its goal. So it's still like you can see just a lot of cases where the automaton still lives close to its original lifetime, even after the perturbation. Now, oftentimes the underlying details change, but the point is that then you wouldn't actually want to go in and treat the organism again with another perturbation, because it adapted a way to, despite changing the environment, to still achieve its goal. So it's kind of like a model for like iatrogenics, or like when a doctor intervenes, like if it's basically like, okay, the doctor sees some details changing in the underlying pattern, but you never know if that's like a method that the organism adapted to live a certain lifetime, or whether that's going to be a disease. And so if you're just, you know, placing a bet based on based on this lifetime graph, if you were just if you knew nothing about the organism, you would you want to default to not doing anything, because it's evolved to try to figure out it for itself, if that makes sense.
Right. And, you know, the more I think about, I mean, I extrapolate this to other systems that we call agential or whatever. At the very least, I can just say in biology, I'm more comfortable talking about it. But I think it's, you know, life as we kind of understand it and know it is really more about finding something to do. You know, you're finding something, you know, organisms can evolve and change and, you know, adapt somewhat, I guess, beneficially if they're continuously exposing themselves to things that would normally be a stress to them. Right. And so I think it does say something very deep about life, sure, philosophically, but also like practically in the way that, say, athletes train and physicians may treat people, is that, you know, a lot of times we're treating the body almost like an engine. Right.
Mhm.
And yeah, listen, if you're a surgeon and if you're going and operating on someone's heart in a very restricted setting, yeah, you can look at it as this sort of mechanical device that's like an engine, and you can kind of pick the pieces and put it the way you want to. But I think something very powerful also that nature and evolution is kind of made use of is that you can give a certain stimulus, and if the system is able to understand it or understand it just enough, it can take a lot from that, right? Who would have thought just by moving your arms and legs a bunch that you can have some pretty big differences in your body and your bones and your muscles and all these different parts of your body, right? And something actually I want to, maybe I can talk about it another salon. I can bring it up a little bit now, but a big game, and I'm talking about this because you're interested in sports, is that, a big aspect of being successful as an athlete, which a lot of people would say is sort of at least peak human physical condition, is actually cognitive, right? And I was actually just speaking with another friend the other day about, even if you have some physical deficits as, say, a basketball player where height is a pretty clear advantage, are there other ways that you could evolve to reach the same goal about making your goal, your scores or whatever, without having to have height, right? And then all of a sudden that starts to become a little bit more of a strategic cognitive game. You're going to take certain paths that other people wouldn't take so that you can come at it from a different angle that they're not able to predict. And so I think looking at that principle, I also think that's kind of what this hormesis thing is, right? So like, if you think about, I think I actually gave this example at the Polymath dinner if you were there to hear this. But, I said, let's just say you're right-handed, okay? And you induce some sort of deficit or an injury. Like, let's say, you break your arm, you break your hand or something. And if the body is, if the goal of the body, there's enough of a pressure for it to have to write something or get a message across, well, how does it do that if you're normally right-handed and you write with that hand? Well, if it can't heal the hand, it's going to look at all these other different pathways and reinterpret them under different contexts to make use of that, right? So if it can't, heal your hand back or whatever, maybe it'll find a way to reuse your left hand. So that's something that happens a lot of times when people hurt one of their limbs, the hand that they write with, is they'll have to learn how to use things, brush their teeth, do whatever with their opposite hand if they're not ambidextrous or something, right? And then, let's just say you can't use your left hand. Well, then maybe it'll try to use your shoulders or other limbs, or maybe it'll figure out, oh, maybe there's another way to use your voice to get this message across, or whatever it is, right? And so it's really interesting if you look at this hormesis process as a way of finding problems, finding goals, and then reaching those goals. I think that's really what the kind of big picture idea of this is. And so my next question to you then, Willem, at least per this model that you've worked on, can you think of any really some less conventional ways biology might solve a problem, right? By being able to search the space of possibility that's normally not explored unless it's signaled and stressed to do so.
That's a tough question. Could you ask it in a different, or like say that again?
So if we're sort of saying that stress and perturbation in this hormesis process are all kind of related or even somewhat synonymous with each other, and if we can kind of agree that part of that is that when you're given a stress, it's about how the organism is responding to it, right? So, I don't know how many people have paid attention to like bodybuilding forums and that whole kind of culture for a while, but, I remember reading years ago about how, it really is all about the technique. Like if someone's trying to get stronger, there's not necessarily only one way of getting stronger to lift that thing that you want to reach. And a lot of the specifics are in the technique, the very specific way that you're moving your arm or you're lifting your weight or doing all these types of things, right? So it's like how people will say perfect practice makes perfect, right? And so what I'm getting at is that when you induce a stress or a perturbation, which is basically giving the organism a goal, right? Does that click so far? It's giving it something to do.
I don't see how that's the same thing.
If your organism is trying to maintain itself or grow, it has to be given some kind of stimulus to even be able to do that, right? Because, if you think about like how we actually prompt change in a system, we have to throw something at it. If it's just sitting still the same, just sitting the same the whole time, it has no reason to actually change. You have to expose it to something that would get it to change itself.
Yeah, but
right?
that's if it's a learning, if it can learn, I mean, it's not clear that our thing, if we give it some perturbation, it's it's not going to like learn from the perturbation and then try to adapt around it. Oh, maybe like if you do that many times, and that's a model for evolution, but it's not like a neural network.
Well, I would actually say that hormesis is kind of like a learning process, a less kind of conventional way
Yeah,
that
but
we.
I'm saying that our perturbation isn't necessarily a learning.
Got it. Again, then I guess my next question would be, why cellular automata in the first place? Why is that? Why does that seem to be such an interesting way of modeling things? I mean, maybe this is a question for both you and for Stephen, but why is this sort of model, this understanding of cellular automata
Hmm.
showing up in you know everything that Wolfram is doing and whatever? But why is that so interesting? And why does that seem to have a lot of use cases and at least potential? Right? What is it about the structure, the dynamics of cellular automata, whatever, that seems to have a lot of generalization ability for things that we want to study?
One, it's like a pretty broad idea, just like evolving, updating some set of rules. Like there's many possible cellular automaton, but it's like a big category. But also, I am also surprised that I just think that it's a really underexplored idea, because most of science tries to use mathematics, and so because of that, I think there's just a lot of low-hanging fruit about nice models that you can make with these systems.
Yeah.
All right.
Much more work on this. Please send it my way.
Thanks.
Have you
Yeah, share this around.
I was wondering if you could maybe like try something like this with like maybe like a convolutional cellular automata instead of a rule-based one. I'm not sure if that would like have like if that would be meaningfully different, or if that would even translate well.
I actually don't know what the difference between like a neural CA, a convolutional CA, but I know that people have done like morphogenic models for using neural CA. So like trying to grow like I forget the name of the lab, but basically, yeah, using neural CAs to model morphogenesis. And it's able to do like much more complicated patterns than the ones are not complicated in the sense of details, but like the shapes that it can form are, more complicated.
Do you have any questions, Willem? Anything that's kind of running through your mind right now? Because I also know you're interested in sports, and I think bringing the field of computation and mathematics into sports could be very interesting in many ways, because it is kind of really all about coordination and stuff like that.
I mean, again, like what I'm wondering about is like, well, one thing is like, how does this idea of adaptive systems in biology, how similar is it to the way that humans learn, like become experts? And I suspect that a lot of the principles that we're finding here are the same, like are similar to what's happening when humans learn things. So for example, like one of the conclusions that from our this is from Wolfram's original post on evolution, is that like there's many different ways to achieve the goal, and if you run the experiment many times, it'll come up with kind of a different way every time. So this is like just different kind of like branches of the tree of life. And I suspect that, and it's kind of similar to what you were saying, Addy, which is that there's many ways to achieve a goal, and that and so this, I feel like it's the same with when you're trying to learn something, where it's like if you're trying to learn chess, for example, it's like the way the algorithm that people use, which is like doing puzzles and playing games and all that stuff, is similar for everyone, but the actual details of the computation involved, the model that your brain is coming up with to get better at chess, is different for each person. I don't know if that's the right example, but I think you guys think it makes sense. So that's one thing that I'm wondering about. Yeah,
because whenever we come up with these theories and these ideas, which is obviously it's plenty of fun, but also I do like to kind of have my head set on things that are very practical, doable applications, things that we can actually test or try out, right? And so one of the things that I've spoken about before that interests me a lot is vision improvement, right? And vision improvement for a lot of different things, I think, for everyday life, but then also specifically for people, say, in sports, people who are athletes. And just like what I was saying earlier, I think there's a lot of things that are cognitive, right, that are actually a part of athlete success. And a lot of athletes, like top athletes, actually apparently have very good eyesight, and they even train for this, right? And so something that I've been interested in for a while is this question, right? So do you see with your eyes or with your brain?
Yes.
Anybody wants to chime in? Yeah, or if anybody wants to chime in, do you see with your eyes or with your brain?
I would say you see with your eyes.
I said I disagree. I think the quality of seeing occurs in your brain because of predictive processing and other reasons.
Yeah, I mean, it's sort of both, but I would more comfortably say it's your brain that's actually doing the seeing, and bringing something actually there. And the interesting thing is you can close your eyes, you can deprive yourself of visual information or whatever, and your brain will create all these very complex visuals at times, right? So people that are doing sensory deprivation tanks, they're meditating, they have closed eyes visuals, they're dreaming, whatever, you're already generating very vivid imagery, when your eyes are closed. But also the other thing that I thought about is, when people misread something or they missee something or whatever, right? Is it something in their eyes, or is that their brain that's doing that? And eventually I kind of came to the conclusion that it could be, brain related, related to the mind and cognition. And so something that I've been interested in for a while is that, okay, if we can kind of make an agreement and have a consensus that vision is kind of taking place in the brain, why are basically all of our interventions to improve at least human vision touching the eye, sometimes even quite invasively, but nothing in the brain? And so I was like, okay, are there ways that I can maybe non-invasively, stimulate the brain to improve vision, right? And now I do want to sort of make a point about this and say that it's not necessarily, if the information is not coming in through your eyes as sort of the lens to the camera sensor, there is kind of technically a threshold, right? And the brain is just trying to make sense of that. And so something that I kind of picked up on is that, okay, using this hormesis idea, if you, in a patterned way, reduce your perception of contrast, edge sharpness, and saturation, is that your brain will try to compensate. It'll try to overcompensate for the loss of expected information, right? And so what that basically means is, like, have any of you ever picked up a really, heavy weight for a while and then picked up your phone and your phone felt lighter? It's a very similar process, right? It's sort of this, there's that very short-term, immediate, adaptation, and then there's long-term adaptation. And I found that by, wearing lenses that do this, if I just, as long as I engaged my visual attention, so if I was reading something, I was playing a game, whatever, I would notice very clear, pun intended, improvements in my visual clarity. I even started doing like distance visual testing and all of that. And I found out that, okay, there seems to be like there's something there to explore, right? Now, obviously, this will need to be looked at and tested more before I can say anything, conclusive. But I think it is interesting that you can bring this principle over to some very less conventional parts that we'd study, because normally people wouldn't think about looking at hormesis as something informational and to the way the brain is perceiving things, right? And this principle that I'm talking about for the mind, for vision, is also applicable to hearing and taste and smell and all these other things. And that's why my kind of, at least big picture idea with this is that this may be something fundamental about information, about the way systems perceive and respond to information in the first place. Like, and the key to this though is that, just how I was saying earlier, is that you want to dose something that would normally be, impossible or hurtful for the system, is that if you blur it too much, then it doesn't work. It has to be just blurry enough where there's still something for your brain to reference and to grab onto. If you don't do that, then the effect doesn't come, right? And so that's why I'm just wondering, this kind of model that you and Stephen have worked on here, about what other things could this be applied to, right? That we just haven't thought of before. And maybe the model is good for figuring out those things that we haven't applied things to before. So I just wanted to hear your thoughts on that.
Yeah, I mean, it reminds me basically like the question, I feel like one interesting question is, what is the minimal model for intelligence? And and like the minimal model for learning. And I mean, Stephen Wolfram looked at this a bunch in his minimal models for, I mean, he did a post on it, minimal models for machine learning. And I mean, again, I mean, in the post, he finds that basically a lot of what's happening when things learn is a similar process to what we see with our like evolution experiments, which is that, you know, they're putting together bits of irreducibility in order to achieve some goal. And so the consequence of that is it's not always clear. You can't always explain what's happening inside the learning system.
I
Okay.
think you should look at attractors and other sorts of systems. I think that would be really interesting, and see if you can get some sort of similar results and ideas. Because there's other systems like network dynamics that actually do map neatly onto real diseases and things like that, kind of like the paper I sent. And so I think there could be some interesting stuff there.
Yep, I'll take a look. Thank you.
All right, cool. All right, well, if there's no other questions, thank you everyone for joining today's Polymath Salon with Willem Nielsen from the Wolfram Institute. I'll hopefully have this up live on YouTube pretty quickly. I don't think we'll be doing a salon for a little while now. I may do one on tulpomancy either later this week or the next week. But in the meantime, you know, feel free to recommend any topics that you think we should cover, guests that we should have on. And yeah, this is gradually picking up, which is a lot of fun. And yeah, I guess I'll see you guys for the next time. Take care. All right, bye everyone. See you. Bye.
Thanks for putting this on, Addie.
Of course. Thank you.
Yeah. Thank you.